Welcome, and thank you all for having me. It's such an honor to present at one of the only comprehensive cancer centers in the mountain region. So again, it's just such an honor to be invited and to be able to speak to all of you today. So first of all, I'd like to get a couple acknowledgments out of the way because without these people, my project wouldn't have been possible. And pictured here is my PhD advisor, Tom Aintradoki. And this project was kind of his brainchild, and I just took it on and ran with it. So definitely a big thanks and acknowledgments to him. And then the second acknowledgement I would like to make is to all of my research animals. A lot of people overlook that most of the time when you see a data point, there used to be a little life behind it. So I just always like to make sure that we give the utmost respect and gratitude to these little rodents that we use all the time. So that gets me to my talk, exploiting a type 3 interferon response to improve chemotherapeutic efficacy. So my project mainly focuses on nanomedicines, chemotherapeutic nanomedicines. And to understand kind of why I'm looking at this first, we need to first understand why nanomedicines are even used to target tumors in the first place. And that is because of something called the enhanced permeability and retention effect, or the EPR effect. So this effect is characterized by an enhanced ability of the tumor cells, especially the endothelium, to just gobble up basically anything that's in circulation. So it gobbles up nutrients. If there's, nanoparticles floating by, it gobbles all those up. And then it has an enhanced ability to retain all of those nanoparticles. So it's eating up more nanoparticles than healthy tissue, and it's holding on to them longer than healthy tissue. And there's a variety of different ways that the tumor cells can take these up, some of which are gaps in the cells, active uptake, and tight junctions. So the problem with chemotherapeutic nanomedicines is that although they do preferentially accumulate in unhealthy tumor tissues, still, there is only about 1% of the injected dose actually accumulates in the tumor. So that obviously leads to other problems in the body. And the main reason for this, these off-target accumulations is the mononuclear phagocytic system, and otherwise known as the reticuloendothelial system. And that includes the liver, the spleen, the lungs, the kidneys, basically all of the major filtering organs. And the main purpose is to just clear the blood of junk, and nanoparticles look like junk to the MPS, so they get cleared rapidly by these organs. Because of this rapid clearance, it leads to off-target toxicities, and this is a major dose limiting factor for most chemotherapeutic nanomedicines. So the primary driver of these toxicities isn't necessarily that the drug is getting in and causing a problem. It's actually immune reactions to the nanoparticles themselves. So it might not be the drug, but it's the package, the nanoparticle package, that is activating an immune response in these off-target locations. So for the longest time, the nanoparticle delivery has been focusing on, stealthing or silencing their nanoparticles so the body can't recognize it as an antigen or a foreign body. And until recently, until the COVID-19 pandemic, it has been a major focus to avoid any immune activation. But because of the COVID-19 pandemic and companies like Moderna, we now have mRNA vaccines that are lipid nanoparticles with mRNA in them. And so activating immune reactions, activating positive immune reactions, is now a primary focus of the nanoparticle field. And because of these advances, we now may be able to immunostimulate the body to enhance or even improve drug delivery rather than have it be a negative effect. So, one possible positive immune response that we might be able to initiate that could alter drug delivery is something that's been overlooked for quite a while. It's called the gene therapy refractory response. Now, this refractory response is observed upon repeated administrations, either, intranasal sprays or injections. But either way, if you give a repeated dose of lipid nanoparticles, complexed with gene therapy vectors, also known as lipoplexes, something strange happens. This refractory response, how I like to kind of imagine it or characterize it for people is, let's say you come and give a dose of your gene therapy nanoparticle, and you come back 24 hours later and you give a second dose. Now, with a regular drug, on the first dose, let's say you can expect 10 micrograms to accumulate in the liver. And on the second dose, you would expect there to be 20 micrograms in the liver. And after a third dose, 30 micrograms. After a fourth dose, 40 micrograms, and so on. But with these gene therapy vectors, once you inject after the first administration, there's 10 micrograms of accumulation in the liver. And then you give a second injection, and the total accumulation is still only at 10 micrograms in the liver. So you come back and you give a third injection, and now the total accumulation is actually down to 9 micrograms in the liver. So it's almost as if these second injections haven't even happened. And in some cases, it actually reduces the accumulation in these target tissues. So this is obviously a problem for gene therapy delivery, but the decrease in accumulation could possibly be used for drug delivery. And one possible explanation for this reduction or this reaction to these gene therapy vectors is that it's actually an antiviral response. And so you're asking, well, why do you think it's antiviral? Well, if you kind of think of the characteristics of nanoparticles complexed with nucleic acids, you begin to see how they could very much look like a virus, because viruses and nanoparticles are both on the nanoscale. They both contain nucleic acid components, and most of the time nanoparticles are coated with PEG or some other polymer, and so are viruses. Viruses are coated with polymers or other sugars. So it's pretty, you can hear my kitty in the background, but it's pretty obvious that the body, when it encounters these lipoplexes, might be thinking, oh my gosh, this is a virus and we need to prevent this from accumulating. So I hypothesized that it was the nucleic acid component of the lipoplexes that is the primary driver of this refractory response. And so to first test this, I wanted to see if I could replicate this refractory response with just naked liposomes. So a lipid nanoparticle without any nucleic acid in it. So I wanted to make sure that this wasn't a saturation effect or just an offhand effect of the lipid nanoparticle itself. So what I did is I treated mice with either an injection of PBS or an injection of liposomes. And then 24 hours later, I gave an injection of dextran. And dextran comes in different molecular weights and sizes. And at about 150 K, it has a hydrodynamic diameter of 100 nanometers. So it mimics the same size as our liposomes. So what you're seeing here is that when you give a pretreatment with liposomes and then inject your fluorescent dextran and then extract them from all your organs, there is basically no difference in accumulation between the control and the liposome. The only difference is in the spleen, and there's actually an increase in accumulation. So we're not seeing any refractory response. We're not seeing any decrease in accumulation. And there's no effect to these liposome pretreatments. So the liposomes do not create the refractory response. So next, I moved on to lipoplexes. I took the exact same liposomes, but I complexed them with nucleic acids, and I did the same treatment. So I pretreated with either a control PBS or lipoplexes, and then 24 hours later, I gave a dose of dextran. And what you're seeing in this data here is there is significant increases in the primary MPS organs, which are the spleen and the liver. So there's this significant reduction of dextran in the spleen and a significant reduction of dextran in the liver. And we also see trends towards reduction in the heart and the kidney. So what we're seeing here is that when you add in this nucleic acid component to the lipoplex, it dramatically alters the accumulation profile of the subsequently injected nano-sized dextran. So to move on and see if this worked on actual nano-sized medicines that we have in the clinic today, we did the exact same treatment, but instead of giving fluorescent dextran, we gave a dose of Doxil, which is liposomal encapsulated doxorubicin. So what you're seeing with this data here is that after a lipoplex pretreatment, we see significant reduction in doxil accumulation in the spleen, in the lung, in the kidney, the heart, the skin, and a trend towards reduction in the brain. Now, you're probably looking at this central graph here, the one with the liver, and it shows an increase in accumulation after the lipoplex pretreatment. And that is most likely due to the pegylation that we had on this lipid nanoparticle. So as I mentioned before, nanoparticles are usually coated in some type of polymer, and that polymer is usually PEG. Now, the unfortunate thing with that PEG is that it can be pretty immunogenic if the body is exposed to it repeatedly. And Doxil is really pegylated. It has a ton of PEG all over its surface. So we thought that this increase might actually be due to an immune reaction against PEG. So we took that away. We gave a pre-treatment of non-pegylated lipoplexes, and again, we rescue that refractory response in the liver. So we, again, with non-pegylated lipoplexes, see a reduction in the spleen, liver, lung, kidney, heart, and skin. Again, proving that the lipoplex complex of lipid nanoparticle with nucleic acid is driving this reduction in the accumulation of the subsequently injected nanoparticle. So the interesting thing about this data was that when we pre-treated animals that were tumor bearing with lipoplexes and then gave a dose of Doxil, the plasma concentration and the tumor accumulation both significantly increased. So you see on the left, the concentration of Doxil still in circulation is significantly higher than the control. And then if you look on the far right panel, you will see that the dextran accumulation in the tumor, as well as the doxil accumulation in the tumor, show a trend towards increase and a significant increase for the dextran. So this refractory response is limiting accumulation in healthy tissues, increasing circulation time, and increasing the tumor accumulation of the subsequently injected particle. So what this is showing is that if we could initiate this refractory response prior to giving a chemotherapeutic nanomedicine, we could ensure that it accumulates preferentially in tumor tissues and reduces accumulation in healthy tissues like the liver, spleen, and other organs, potentially reducing side effects. So one explanation for why the tumor has such a different accumulation profile to healthy tissues is the immunosuppressed microenvironment, or just the dysfunctional microenvironment of the tumor in general. And what you're seeing here is that when we give a dose of highly immunogenic lipid nanoparticles, you're seeing the interferon production or just inflammatory cytokine production in the liver or the tumor. And what you're seeing after a first injection is that the liver has very robust cytokine production and the tumor has a little bit cytokine production. But when you come back and you give a second dose of these inflammatory nanoparticles, you see there is a huge increase in the liver cytokine production, but there's almost nothing in the tumor. So the tumor is not reacting immunologically or in any kind of inflammatory way to these lipoplexes. And because of that, we think that it's suppressing whatever immune response is driving the refractory state and allowing the tumor to continue on just gobbling up nanoparticles like it normally likes to do. So, how exactly can an immune response to these lipoplexes somehow significantly alter nanoparticle distribution of a completely different nanoparticle that's subsequently injected? So, what's going on here? How is this happening? And to understand how this is happening, we first need to understand how nanomedicines even get into the cells or tissues. And the primary mode of transportation or the highest correlated mode of transportation for nanoparticles, as you see in this graph here, is at the tight junctions. So the Spearman correlation is just showing how many nanoparticles were associated with these structures in the cells. And as you can see, the tight junction has the highest correlation, the highest association with the nanoparticles, suggesting that tight junctions play a significant role in nanoparticle entry into the tissues. So you're probably looking at this static barrier, this blob between these two cells and thinking, well, how the heck does a nanoparticle get in between that static barrier like that? Well, it's actually not static at all. You know, a lot of textbooks and a lot of representations of tight junctions just kind of show this bridge, this like stopgap between the cells, and it's just two linkers together, but that couldn't be further from the truth. So what you're seeing here in these panels is actually tight junction protein networks that are linking together and unlinking. So hopefully you all can see this video playing, but it's an incredibly awesome video of these proteins just breaking apart, forming back together, breaking apart, forming back with another part, and just continually making these loops and circles and just continually changing. And so with this in mind, you can kind of see how a nanoparticle could, sneak into one of these loops when it breaks apart. And then when the other side breaks apart, it can go to the next chamber and the next chamber and the next chamber. And that's basically how nanoparticles can move through these tight junctions. It's just a web that's continually forming and reforming. So, since we know that tight junctions obviously play a role in nanoparticle entry into tissues, they actually may represent an excellent target for modulating nanoparticle delivery to tissues. So it's been well established in the literature that antiviral responses and other immunological stimuli are actually quite potent quite potent effectors of the permeability of tight junctions. And one of these interferons or immunological stimuli that has recently been described is the type 3 interferon or interferon lambda. And this interferon lambda has been shown to be directly involved in increasing tight junction protein co-localization and tightness. So what exactly are these interferons? There's the third family of interferons, so it's not alpha or beta, but this is specifically lambda, and there are 4 lambda subtypes that are active in humans. And these lambdas are rapidly produced in response to pathogen exposure or viral exposure. And the primary role of these interferon lambdas is to actually regulate the barrier functions of both epithelial and endothelial cells, while also inducing a broad set of antiviral genes without stimulating excessive inflammation. So that's the main difference between these interferons and the interferon alpha, beta, gamma, the other inflammatory ones, is that these ones do not stimulate inflammation. In fact, they are more of a resolving interferon. Excuse me. So because of this, I wanted to show you some data from a recently published paper showing interferon lambda's direct effects in regulating tight junction tightness and co-localization. So what you're seeing on the left are 3 different panels. The first panel on the left is a measurement of the barrier tightness. So tier is basically just how resistant is that barrier to things going through it. And what you see is that in the red dots, when it's treated with interferon lambda, the barrier resistance increases significantly when compared to control and an inflammatory cytokine like interferon beta. And what you see in the middle panel on the left here is when you infect a cell with salmonella, which causes it to lose its barrier integrity, but then rescue it with interferon lambda. Interferon lambda significantly rescues the barrier resistance when compared to control or interferon beta. And the last panel on the left here, we show the amount of infiltration of a fluorescent probe into the tissues after salmonella infection. And when you rescue the salmonella with interferon lambda, it shows that it can prevent the infiltration of this fluorescent marker into the tissues. Moving on to the right panels, it is a visual representation of basically what we just saw in the left panels. If you look up in the very left-hand set of squares, you'll see the green lines and the red lines. The green is ZO1 or zona occludin 1, which is the primary tight junction protein. And then you'll see the red lines, which is Claudin 5, another important tight junction protein that regulates the tightness of those. And this upper left-hand panel is the control, and you see this nice kind of yellow overlapping red-green border in these nice healthy control cells. Now, moving on to the right set of panels, when you treat it with interferon lambda, you can see all of those proteins snap together. It's almost a perfect alignment of red on top of green, so you have that super bright yellow border in between the cells. Now, if you treat these healthy cells with LPS, moving down to the bottom left panel, you see that the zona occludin, it's still there, but it's kind of fuzzy, it's mixed up. But then the Claudin 5, it's just all over the cell. There's no organization to it whatsoever. And if you look at the overlapping, all you can see is the ZO1 that barely defines these cell borders. But if you come and you rescue these LPS treated cells with interferon lambda, those proteins snap back together and you have that nice yellow alignment over the cell borders. And in the bottom panel, that's just showing the exact same thing, but with live blood-brain barrier cells. So you're seeing this in vivo live time. After you rescue with interferon lambda, it snaps all of those tight junction proteins back together, increasing the barrier integrity. So with that information in mind, I hypothesized that the refractory response is actually an antiviral response to the lipid nucleic acid complex, and the antiviral stimuli behind it, driving it, is interferon lambda. So to test this hypothesis, I treated animals with either a control PBS or dextrose injection, a liposome injection, and these liposomes have no nucleic acid, or a lipoplex treatment. The lipoplex is the same liposome, but with nucleic acids complex to it. So what you're seeing in panel A is the overview, and the only treatment that initiated an interferon lambda response in the plasma were the lipoplexes. So this shows that after you give an injection of lipoplex, at about 12 hours, you first start to see the interferon lambda. It peaks at 24 hours, and then it rapidly declines at about 36 hours. And so this is most likely due to the fact that the lipoplex is exposed to the body recognizes it, and then it's immediately cleared. Once it's cleared, the interferon lambda comes back down. So this aligns really well with its role as a first line defender or a resolving interferon, because if you had a full blow-in reaction, you'd expect there to be a steady level of this inflammatory cytokine, but instead you just see a quick blip of the interferon lambda and goes back down. And so these other panels are just showing that the liposome, non-nucleic acid treatment, and the dextrose treatment, they do not produce a detectable level of interferon lambda, showing that it is in fact the nucleic acid portion of the lipoplex that is triggering an antiviral interferon lambda response. Oops. All right, so to verify that lipoplexes and interferon lambda were in fact initiating a response in epithelial cells and endothelial cells, I first worked with human lung microvascular endothelial cells. Now, I chose these cells because it makes sense that if you breathe in a virus, the first place you might want to restrict it from getting into the body is in the vasculature of the lung. So that's why I chose these lung endothelial cells. But when we treated them with interferon lambda, there was no response. That we didn't see any relative change in expression of the interferon lambda genes. And that's most likely just because working with endothelial cells in culture is extremely variable, and their gene profiles change almost immediately once they've been harvested and plated. So we didn't really rely too much on these lung experiments, and instead we moved on to human liver epithelial cells. And the reason why we chose the liver epithelial cells is because the liver is the primary MPS organ, the primary organ that's going to be clearing nanoparticles from the circulation. So we wanted to see if interferon lambda and lipoplexes were going to initiate a reaction in the primary organ of nanoparticle accumulation. And what we see is that after treatment with interferon lambdas 1 or 2 and 3, we do see a significant increase in the expression profile in these cells. And even more so when we treat with lipoplexes, it has the most robust response. So what we're showing here is that interferon lambda can in fact act on these epithelial cells, and they can act on the primary epithelial cells that will be doing the off-target accumulation of nanoparticles. So to see if, how does this response, how does the interferon lambda response, if you just, deliver something to the lungs, how does it go systemic? Or if you just give like a sub-Q injection of a vaccine, how does that response go systemic? So we wanted to see if liver cells that produced interferon lambda in response to lipoplexes were capable of activating other cells that had not been exposed to lipoplexes. So what you're seeing here is liver cells that were treated with 1 microgram, 0.5 micrograms, or 0.25 micrograms of lipoplexes. And what you can see is that after a treatment with 1 microgram of lipoplexes, there's a significant production of interferon lambda by these liver cells. And then I took the media from those liver cells and I put them onto untreated liver cells, and I looked at the gene expression change in those cells. And what you're seeing on the right table here is that when I took these untreated cells and put interferon lambda that was produced from other liver cells on them, they did in fact react, they did change their gene profiles. So that means that even if one little lipoplex gets into the body and it initiates an interferon lambda reaction, it's going to be able to go systemic, because that liver cell is going to tell its neighbors that, oh, hey, there's a virus invading. It's going to tell its neighbors, it's going to tell its neighbors, and eventually the whole body is going to have this interferon lambda refractory response to the lipoplexes. So we wanted to see if just a direct administration of interferon lambda would cause the same type of refractory response as a lipoplex injection. So, let's remove the middleman. Let's just see if interferon lambda has the same effects as a lipoplex. And what you're seeing in these graphs here is I gave mice either a pretreatment of PBS control or 1 microgram of interferon lambda intravenously, 10 micrograms of interferon lambda intravenously, or 10 micrograms subcutaneously. And with every interferon lambda group, we see significant decreases in the skin, in the heart, in the kidney, the lung, the liver, and even a significant decrease in the spleen. So again, we have shown that interferon lambda is capable of reproducing this refractory response. And this kind of proves the theory that the lipoplexes are initiating an antiviral response. And this proves that interferon lambda is the cytokine that is driving that response. But I want to point out this first panel, the skin panel. This is particularly interesting because the primary site for off-target toxicities of Doxil is in the skin. So if we give a subcutaneous interferon lambda injection before we give a treatment with Doxil, we're going to have an extremely significant reduction of Doxil in the skin, which could potentially lead to reduction or even elimination of the primary off-target toxicity of Doxil. So that's why this first skin panel is so interesting. Now, to make sure that this, again, would have the same type of refractory response in the plasma and the tumor as lipoplexes, we just looked at accumulation in plasma. And what we see again is that at sub-Q 10 micrograms of interferon lambda, there is more plasma, or there is more Doxil in circulation for a longer amount of time. And we do see a trend towards increase in the tumor over time. And even out to 72 hours, there is still that increased accumulation in the tumor. So we've potentially reduced off-target accumulation and toxicities while we've increased circulation time and increased tumor exposure. So to tie this all together, we wanted to actually do a treatment model. You know, we've proven that this can reduce accumulation and everything, but how does it actually work when we're treating animals with cancer? And what you're seeing in these three panels, the first panel on the left is the survival study. So right away, you can see that our control group, the black circles, the ones that were treated with interferon lambda and Doxil, had the highest survival rate. The animals that were treated with either just control interferon lambda, or control Doxil, all perished before day 15, while almost half of the animals that were treated with the interferon lambda Doxil were still alive. Now, how efficacious is this on the tumor growth profile? Well, the center graph here shows you our dosing schedule and the growth of these tumors. So we started the tumors, and once they hit about 150 millimeters cubed, we started our dosing. And what you can see is, again, the black circles, the interferon lambda plus Doxil circles, have the lowest amount of tumor growth, and it's reversing it, it's reducing it more than any other treatment, and it continues to keep that tumor growth reduced well after the last dose. So it's much more effective, much more efficacious in keeping the tumor volume down and keeping growth from moving forward. But the best part about this, the real bread and butter of this treatment, is we already know that Doxil can treat tumors. We already know that it's a relatively efficacious treatment. But with an interferon lambda pre-treatment, what we can see here is this is the animal body weight change. So our interferon lambda plus Doxil treated animals, the black circles in the middle here, they stay steady. They don't have a reduction as significant as just the Doxil treated animals. And in fact, just the Doxil treated animals had such a massive body weight loss that they had to be euthanized. So it wasn't necessarily the cancer that got these animals, the gray squares. It was just the Doxil, just the toxicity of the Doxil that ended the study for them. However, if you give the interferon lambda pre-treatment, these mice can handle it like a champ and continue to live on and continue to maintain that body weight without having that toxic side effects of Doxil continue to affect them. So now that we know that interferon lambda is the primary driver of the refractory response, and that we can use that to potentially increase the efficacy of Doxil, Just for shits and giggles, we wanted to see, could this work on a small molecule? And we kind of thought, this is a throwaway study. It's probably not going to have an effect because small molecules are obviously much smaller than nanoparticles. But what we saw is that in some organs, after a interferon lambda pretreatment, we can even reduce the accumulation of a small molecule like doxorubicin. There was a significant reduction in the spleen accumulation, significant reductions in the lung, kidney, and liver. So this refractory response may also be able to affect small molecule accumulation. If it can reduce the side effects for a nanoparticle, it could potentially reduce the side effects for these small molecules like this. And one explanation for this data is that doxorubicin is very highly protein bound. So it's possible that when you give an interferon lambda injection, it creates this refractory state that prevents proteins like albumin that are bound to the drug from getting into the tissues as easily, possibly reducing the accumulation of doxorubicin in those tissues. So that's just a hypothesis, and this is just cool data. We haven't really progressed with this, but it's just very interesting to see that this interferon lambda can, in fact, alter the distribution of small molecules. So to summarize what I've shown you about my research is that exposure to lipoplexes initiates an antiviral refractory response, which can significantly alter biodistribution of other nanoparticles. This refractory response is dependent on the presence of nucleic acids in the nanoparticle formulation, and this response is in fact an interferon lambda response. With this interferon lambda, we can give a pretreatment that can significantly reduce the off-target accumulation of doxil while also increasing its circulation time and potential tumor exposure. This interferon lambda pretreatment can significantly reduce off-target accumulation of a small molecule as well. It's possible that a pretreatment interferon lambda could increase the efficacy and safety of both doxil and doxorubicin. So a little bit of perspective on this. Interferon lambda has been extensively studied in patients with hepatitis. There's been over 20 clinical trials with over 3,000 patients. And while those drugs weren't approved for hepatitis because the patients that had hepatitis showed a little bit of liver toxicity, if the patients were healthy, there were no toxic side effects with interferon lambda. So it could potentially be used to treat something as long as your liver is healthy. And in fact, that is exactly what we saw is during the COVID-19 pandemic, using pegylated interferon through a sub-Q injection, successfully finished phase 3 clinical trials. So this could be on the market in the near future. It's clinically available, it's safe, it's effective. So an interferon lambda pretreatment is, it could be possible in the very near future. So due to the interferon lambda's dramatic effects on both nanomedicine and small molecule biodistribution, I think it's important that we continue to investigate the interplay of antiviral responses with drug delivery, because we all know that inflammatory responses and other pathophysiological states can alter pharmacokinetics, but not a lot of people are looking at antiviral responses or those type of cytokines and how they might alter drug delivery. So we need to focus on that in the future. Just to add in here, I am currently in a different lab now, and we're also focusing on chemotherapeutic side effects. And the primary project that I'll be helping on is immune checkpoint inhibitor mediated nephrotoxicity. So basically, patients that are prescribed these immune checkpoint inhibitors can develop some pretty bad acute kidney injuries. And we are just trying to find some early biomarkers that can tell us when exactly a patient might be at risk of developing these injuries and how we might be able to mitigate them. So yeah, there's a clinical aspect. We'll be taking samples from patients here at the hospital and looking for biomarkers in them, and then we'll simultaneously be running an animal model to, get a closer look at possible injury progression with these immune checkpoint inhibitors. So with that, I'd like to thank everybody in the Antrodoki lab, especially Dr. Tom Antrodoki, my advisor, Dr. Madison Rico, Jamie Betker for providing some of the data, and Dr. Emily Heeman of Ohio State University, who was on my committee and a big reason why we have all the information on interferon lambda. And I'd also like to thank my current lab members, Dr. Melanie Joy, Lauren Thompson, and Sarah Asby for providing those final slides. And of course, our NIH funding. So I'd also like to thank my family and friends. And with that, I will take any questions that you guys have.